I don't know what it is about Zoom, but Zoom seems to work remarkably slowly. That they may know you, the only true God. You want one? Then we won't.
Thank you. I can't believe spring break is next week.
Yeah.
Wild. Entire hour, I think it's my time, and I know I'm running out of time, so I'm like my adrenaline or something.
Hey, Dane.
Hi, how are you doing?
Any news yet?
Nope, no news.
Okay, how's she feel?
She's feeling a little bit better, so that's good.
Do
I haven't started to feel anything yet, so.
you think this may be a false alarm?
Probably, yeah.
Okay, let's hope it is.
Yeah.
I have handouts. I'll put the handouts online.
Okay.
Or let's see. Do you have your email open?
Yeah.
Let me try and email them to you.
Yeah, I got them.
Excellent. Okay. Very good. All right. Do you see the slides?
Let me get back to Zoom real quick.
Okay.
Yeah, I can see the slides.
And do they move?
Yep.
Excellent. Okay. We're going to get started then. Okay. I sent Dane the handout so you can follow along with us.
Did you write any questions in the handout? I mean, they may be poorly enough, but...
I'm pretty sure I just used a slide.
Okay, I just wanted to check because I was dedicating a lot
Yeah.
of the handout, so then it's more time.
Yeah. So, yeah. All right.
For the one final question, for the fatty acid, like the beta oxidation, we had two different slides from last semester that you did, or should you use fatty acid oxidation 1 or 2 regulation? Should we just focus on beta oxidation?
We didn't cover beta oxidation. Okay. So you don't really need to cover beta oxidation. You need to be more concerned with. Come on.
Because I know that was a review that we did in the beginning.
Yeah, fatty acid oxidation was to get you ready.
So we should be more concerned about like the making complex carbohydrates and the carbohydrates.
Yeah.
So I'm in group 3, and I was like,
There it goes. There it goes. Okay. All right. The last time we looked at the synthesis of cholesterol. We saw that you start with the thiolase reaction. And we remembered that there's a thiolase that's used during ketogenesis, and there's a thiolase that's used during fatty acid oxidation. This is a different thiolase. So while it catalyzes the same reaction, it is not a mitochondrial enzyme. It's encoded by a different gene. The so-called ACAT2 gene encodes this protein, ACAT2.
And it's in the cytosol, right?
The enzyme, yeah, is localized in the cytosol. So that's how we start essentially synthesizing cholesterol. Remember that you get acetyl coenzyme A to the cytosol via citrate that is shipped to the cytosol from the mitochondrion. Then it experiences the enzyme citrate lyase, ATP citrate lyase, that converts citrate to acetyl coenzyme A and ATP, but you have to expend an acetyl coenzyme A and oxaloacetate, but you have to burn an ATP to do that. Reason being is that you're making a high energy thioester bond. To make that bond, you have to burn an ATP. The oxaloacetate then gets recycled back into the cytosol. Typically, it's converted to malate, because remember, there's a malate dehydrogenase isozyme that's cytosolic. Then the malic enzyme will convert it to pyruvate.
There's a big circle pathway that's on one of our slides.
Yeah, yeah. And then malic enzyme uses NADP. So you reduce this carrier. Also remember malate is 4 carbons, pyruvate is 3. So what else are you going to lose? CO2. And you're going to get a pyruvate, and then the pyruvate gets shipped back into the mitochondrion. So this essentially is just to remind you. Good night. Well, it's all this little stuff, no substantial chalk to use. So this essentially is how you get the and there are transporters in the inner mitochondrial membrane for these. This is all worked out. There's a citrate transporter and a pyruvate transporter. Monocarboxylic acid transporter, and then the mitochondrial pyruvate transporter, MPC. There's an MPC1 and an MPC2. They're encoded by different genes. There's 2 of them. They work about the same. So, and then this is a citrate transporter, so forth. Okay, so from that acetyl coenzyme A, you make the acetyl coenzyme A. That's the cytosolic thiolase. And then you're going to take that and you're going to add another acetyl coenzyme A to that. And you're going to get the beta hydroxy beta methylglutarate coenzyme A, HMG coenzyme A, that's HMG CoA synthase. The high energy thioester bond provides the energy for the synthesis of this molecule. Then you're going to reduce this, and that's HMG CoA reductase. And again, as we said, that is the most highly regulated step in the cell. And this uses two reducing equivalents. And as you can see, you are going to reduce this end of the molecule to an alcohol. And you'll give up a coenzyme A. So that's mevalonate. Once you make mevalonate, you are committed to the synthesis of cholesterol. This is the rate limiting step in this reaction. Now, from the handout, you can see that this enzyme is subject to a good deal of regulation. So there is a kinase, HMG CoA reductase kinase, which you can see on your handout. Yeah, this one, stapled one. And this particular kinase is subject also to regulation by phosphorylation. So if you phosphorylate the kinase, as you can see, it's active. And then the phosphorylated reductase is inactive. The dephosphorylation of the kinase inactivates it, and the dephosphorylation of the HMG CoA reductase activates it. So glucagon, which stimulates the production of cyclic AMP inside cells, activates protein kinase A, and protein kinase A, as you can see, activates a protein kinase that will phosphorylate a protein inhibitor of the reductase phosphatase. That's how increased levels of cyclic AMP prevent the dephosphorylation of the reductase kinase, HMG CoA reductase kinase. I'm on the first page, Jenna. See at the bottom there? Yeah, that's right.
Are they reciprocally regulated then?
Yes.
Okay.
Yes. And insulin does the polar opposite. Insulin will activate the reductase instead of inactivating it. So and it inactivates the kinase, because the kinase, when it phosphorylates HMG CoA reductase, it decreases its activity. So insulin is, whereas glucagon inhibits the phosphatase that takes the phosphate off the kinase, thereby making sure the kinase is more active. Insulin will do the exact opposite. Insulin will favor the inactive form of the kinase. They reciprocally regulate HMG CoA reductase. Now, when HMG CoA reductase is phosphorylated, it's less active and it is also marked for degradation. So its protein half-life also goes down. Let me say that again. When HMG-CoA reductase is phosphorylated, not only does its activity go down, but it's marked for degradation. So the protein half-life also goes down. So you can see this protein is controlled at two levels. It's controlled at protein half-life and it's controlled at activity. Both of those are controlled by its phosphorylation, dephosphorylation state. So as you can see, the same protein that dephosphorylates the kinase, the HMG-CoA reductase kinase, also dephosphorylates HMG-CoA reductase. And when that phosphatase is inhibited, as a consequence of increased cyclic AMP levels, the upshot is that HMG-CoA reductase is less active and tends to be more phosphorylated. So it also tends to have a shorter half-life. So on your paper, please write this: glucagon equals less active HMG-CoA reductase that also has a shorter half-life. Dane, you getting that? This is a little complicated.
You're fine. You said glucagon equals HMG reductase and it's less active and has a shorter half-life.
Yeah,
okay.
Yeah, because of its phosphorylation, dephosphorylation state. When glucagon is present, HMG-CoA reductase tends to be phosphorylated.
Okay.
And so does the kinase. That makes the kinase active. That means that HMG-CoA reductase will be more phosphorylated.
Okay.
All right. We good? Sure. When glucagon is present, HMG-CoA reductase will be phosphorylated. That makes it less active and also causes it to have a shorter half-life. Insulin does the exact opposite.
So insulin makes HMG-CoA more active for longer.
Yeah.
And it's less phosphorylated.
Right. That's why sugar-rich diets can jack up your cholesterol. And you see it right there. Because the enzyme is more active. When it's more active, you make more cholesterol.
I thought for a second you were pointing at the M&Ms. Like, did that have sugar in it?
Yeah, they did, but...
Yeah, so when you say insulin does the opposite, you're comparing it to glucagon, so it would, it would tend to dephosphorylate. Yeah. Okay.
This cat is now in the picture. This cat's very curious. Yeah, insulin does the exact opposite. It favors the dephosphorylated state of HMG-CoA reductase. And in the dephosphorylated state, HMG-CoA reductase is much more active. And it's much less likely to be broken down.
Are you saying also that the kinase reductions are kind of also opposite? Or is that not?
Well, yeah, in its state, because the phosphorylated HMG-CoA reductase kinase is more active, whereas the phosphorylated HMG-CoA reductase is less active.
So if one is inactive, the other one's active.
Yeah.
Okay.
Right. One when phosphorylated is active, the other one phosphorylated is inactive. All right, now we're going to activate these. After we activate them, we're going to link them together. So the activation requires you that you burn 3 ATPs.
And this is also the no turning back point, right?
Yeah, we're at the no turning back point. We're committed to this pathway. Whether we make cholesterol with this pathway is another story, but we're committed to this isoprene subunit making pathway.
ATP is the ring.
Pretty much, yeah. Yeah. All right, so the mevalonate will go through 2 successive phosphorylations by mevalonate kinase. Gosh, I don't like the name for that enzyme. We're going to call it mevalonate kinase, okay?
Is that the one that has two names?
Yeah, I much prefer mevalonate kinase because that tells you what it is. This makes it sound like the phosphate transferring is passive. It's not. It's active. You burn an ATP. Hello?
Mevalonate kinase?
Yeah. Mevalonate kinase and then phosphomevalonate kinase. And you get essentially 5-pyrophosphate mevalonate. So you have 5-phosphomevalonate and 5-pyrophosphomevalonate. So these two enzymes, and interestingly, mevalonate kinase in humans is a peroxisomal enzyme, strangely enough. Yeah, both of these enzymes, mevalonate kinase, phosphomevalonate kinase, are peroxisomal enzymes, which is a little weird, but then we have the pyrophosphomevalonate decarboxylase that is going to activate the alcohol here, but that's simply going to cause electrons to be withdrawn from this carboxylic acid. The carboxylic acid is going to leave. That will shift electrons over here, and you make a double bond. So that's a pyrophosphomevalonate decarboxylase. Yeah, it's also called isopentenyl pyrophosphate synthase, or synthetase, excuse me. But we're going to call it this. Thing is, isopentenyl pyrophosphate is nicely abbreviated IPP. So calling it IPP synthetase is kind of handy, but that's not, this is more descriptive of what it does.
Right.
So I like this better. Pyrophosphomevalonate decarboxylase.
So we should be referencing something that we gave them for all the names.
Yeah, except use mevalonate kinase.
Yeah, it's in the handout.
Yeah, yeah. Because me no like you that other name. Okay, then you get the activated isopremes, isopentenyl pyrophosphate. So you lose the CO2, you lose the inorganic phosphate, two very good leaving groups, one very stable, one capable of extensive resonance structures that stabilize it. And you get these structures where this double bond will flip this way, that way, isopentenyl pyrophosphate and the dimethyl allyl pyrophosphate. And these are your activated isoprenes.
One pyrophosphate melanate carboxylase. Wow. That provides that requires.
Yes. Yes. And as you look at the mechanism, you can see why. That oxygen is not enough to pull the electrons off the carboxylic acid so that it leaves. The phosphate is. It's a much greater electron sink. And so the electrons move, CO2 says, bag this, I'm leaving. Or something like that. Dr. Baldwin would whip me if he heard me say that, because he doesn't like personalizing molecules. Like I said, every party has a poop, and that's why we invited you. So. All right, and these activated isoprene units will then be linked together, and they're eventually going to form squalene. Squalene gets cyclized to the precursor of cholesterol called lanosterol. Lanosterol.
Do we do this like squalene in labs, like solutions that are part squalene?
Squalene is called squalene because it was first discovered in sharks.
No, I just remember seeing it in one of our labs. lab protocols that were like it was like something for that squalene.
It's a good solvent. In fact, squalene is part of the reason sharks float. Sharks don't have a swim bladder. They concentrate urea in their tissues, but they also concentrate a good deal of squalene. When you dissected the shark in high school, I don't know if you did, it's really oily.
Yeah.
That's squalene, mainly. That's why it's called squalene, because squalos in Greek means shark. In fact, Squalus acanthus, that's the name, that's the Latin name of the dogfish shark. Learn something every day. You didn't?
No, my biology teacher was allergic to formaldehyde. I feel like we didn't even use formaldehyde.
You can get other preservatives nowadays.
Yeah, but I don't know why she still didn't do it. But she picked like 3 people in our class to go to other biology classes to it. Sometimes like them, but like the rest of us.
To teach math. I mean, honestly.
Yeah.
Dissecting is what made biology worth it. Getting your hands dirty, getting in, seeing it for yourself.
My first dissection was freshman bio here. First time I ever did it.
If I may tell a short story, there was a teacher at the middle school. He had students summarize the newspaper. He never did any science, so I went and did a science show at his class. My daughter was in that class. The students cheered. I blew things up. I did the sugar and the sulfuric acid to make the worm and all this sort of stuff. And I told him, I said, a lot of this stuff, it's dime store stuff. You can buy it at a hardware store. And I had all my protocols written out, typed out, and I gave it to him. And I said, you can do this. It's easy. It's all explained here. My email's at the bottom. You can ask me any question. This stuff is easy. Easy, easy, easy. You can do this. Here's some YouTube links. You can look at stuff. You watch me do it. I was getting ready to leave, and I noticed the stuff I gave him was in the trash. He didn't improve. He didn't want to improve.
Yeah. Well,
and
it's really annoying because like my chemistry teacher in high school was way better. And so like that's when like when we were roommates freshman year, I hated biology because I had such a terrible experience that I was like, we had off freshman year. She's like, you should be a chem major because you hate bio so much. I'm like, well, I like biochemistry. I just don't like the bio. And so like I've had to learn to love it now. Like, get over it because I had such a terrible teacher for it.
Yeah.
Teachers can make or break you.
They really can. I got into life science because of my bio teacher.
Yeah. Thankfully,
Rex Clark.
Thankfully, the chemistry teacher also taught a like genetics class that I took my senior year, and so that's what got me into it, the biochem realm. And I was like, oh, this seems more interesting. I like it. Yeah, I hated all of it. So I had that teachers that I took an anatomy class. I knew I liked science.
They're telling me about their teachers, Dane. You probably can't hear them, but
yeah, I was an anatomy teacher.
I can hear them a little bit.
Oh, you can hear them a little bit? Okay.
Yeah.
Okay, so.
Yeah.
All right, let's get back to linking up these isoprene units. So you have the isomerase that will flip the isopentenyl pyrophosphate to the dimethyl allyl pyrophosphate back and forth. And that isomerase is actually quite important. And as we said, these are the precursors to these vitamins that are made in plants, also to carotenoids, the phytol chain of chlorophyll, bacteria chlorophyll, phaeophytin, any of those. Rubber is an isoprene, repeated isoprene unit. Quinone, electron carriers, ubiquinone, plastoquinone, all those. Vitamin D is also made from cholesterol. All of those are made from this pathway. So your first enzyme is called prenyl transferase. Prenyl transferase. Prenyl transferase is going to take these two molecules into a head to tail condensation, a head to tail condensation. These groups are activated, so you don't need to use any more ATP. And I think those are on your handout. Okay, so you have the dimethyl pyrophosphate, the isopentyl pyrophosphate, and you are going to go head to tail with that by prenyl transferase, and that gives you geranyl pyrophosphate. That is a C10. The isoprene units are C5s. This is a C10. Right, head to tail. Notice the geranyl phosphate is still activated. It's still activated. Now, you're going to add yet another isoprene unit to the geranyl pyrophosphate, head to tail, and now you get farnesyl pyrophosphate. Farnesyl pyrophosphate. So the prenyl transferase is a rather busy enzyme, as you can see.
So it's the head of the dimethyl attached to the tail of the iso.
Yep. And then the head goes to the tail of the geranyl to make the C15 farnesyl. Some books call this enzyme farnesyl diphosphate synthase. I don't like that name because the enzyme is a multifaceted enzyme that catalyzes more than one reaction. I like prenyl transferase much better because these are prenyl groups that keep getting transferred. So I like the prenyl transferase name better. Okay.
Wait, so what's added to the geranyl?
Another isopentenyl pyrophosphate.
Oh, I see it. I see it. Okay.
Yep. And you can see that reaction here. Once again, head to tail, head to tail. Your next reaction is going to take these two farnesyl pyrophosphates and it's going to join them head to head to make a C30 molecule displacing two pyrophosphates. That is squalene synthase. Squalene synthase. These enzymes are in the smooth ER, by the way. That's a mistake. 15 plus 15 is 30. Yeah, that's a mistake. Scratch that out. Make that a 30. There's a mistake on the handout, Dane. Squalene should be C30, not C20.
Okay.
I did this in ChemDraw, so I can't fix it. I gotta redo this in Marvin Sketch. And that is squalene, and squalene is going to get cyclized to make lanosterol. So this is the final step in this section of cholesterol biosynthesis. Squalene undergoes a two-step cyclization to yield lanosterol. The first reaction, which you can see here, uses the squalene epoxidase, or so-called squalene monooxygenase. This enzyme uses NADPH as a cofactor and introduces molecular oxygen as an epoxide at the 2,3 position. At the 2,3 position, which is right there. 1, 2, 3. You're adding it essentially across the double bond.
Oh.
And then in the second step, the epoxide is converted to lanosterol through the activity of the lanosterol synthase activity of squalene synthase.
So it's just once you break open that epoxide, is it just like a chain that of other double bonds breaking open and forming a ring?
Yeah, essentially. So these electrons go here, these go here, these go here, and what have you. Yeah, pretty much. Exactly. And you can see essentially how once you withdraw these and go over here, this then is this, that's going to fall. It's all going to, yeah, it's a cascade essentially. In humans, even though these two enzymatic activities form squalene synthase, they are encoded by two separate genes, but they form a complex in the ER that forms so-called squalene synthase. And that gives you lanosterol. In plants, you have the conversion of lanosterol to stigmasterol and cytosterol. You don't make cholesterol in plants. You do, but it's short-lived. It's an intermediate. So if you eat something that grows in the ground, you won't get any dietary cholesterol. So I have to be careful because I'm going to show you a pathway that'll have cholesterol in it, but it's only as an intermediate. No. In fungi, you take lanosterol and you make ergosterol. There's no cholesterol in fungi either. So if you saute some mushrooms tonight, there'll be cholesterol maybe in the butter, unless you use, I can't believe it's not butter, which doesn't have cholesterol. But that won't have cholesterol either, that'll have ergosterol. Animals, however, make cholesterol from lanosterol. So all three of these organisms make lanosterol. It's what they make from it. The end product, that is different. All right. Yep. They still make lanosterol. And typically in this class, I would stop right there and just say, and a miracle occurs, and then you get cholesterol. I'm not going to do that this year because, one, some of the diseases that are interesting actually affect this later part of the pathway. And then also the biochemistry of some of these reactions is really interesting. It truly is. So, all right, so now we're going to move to this handout. Now,
This is how you get cholesterol
from lanosterol. Now there's a couple ways to skin this cat. I know Amanda doesn't like that metaphor because she likes her kitty. But there's two main pathways to get from linosterol to cholesterol. It seems as though in most cells, you take those two pathways and use half of one and half of the other. That's what this is. So there's two pathways. I'll show you them and we'll go through them. And just the conversion, blah, 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 and yeah, the other steps and what have you. Oh, also, we're going to talk about yeah. Anyway, we'll just
Get that when we get to that.
yeah, pass that bridge when we get there. Okay, so the first stage uses an enzyme that's called C14 sterol demethylase. C14 sterol demethylase. And it is encoded by the CYP5-1A1 gene that you can see there. Remember, all these genes encode proteins that are embedded in the smoothie R.
Should we know what genes encode it or the name of the protein?
Both.
So what was the, can you write down then?
CYP51A1. So CYP51, that's a whole class of cytochrome P450s, and then it's the A1 member of the number 51 branch of the cytochrome P450 family.
So what does that encode?
This encodes the C14 demethylase.
So you have the CYP1A1, but not other things.
Right. The C14 sterol demethylase is what it does.
C14 sterol
demethylase. And so the arrow, see the arrow, where the arrow is? That's carbon number 30, but it's attached to carbon number 14. That methyl group goes bye-bye. And you'll notice it's turned into a formate. Now, if it's turned into a formate, what does that tell you? This is a mixed function oxidase. It takes molecular oxygen. So it converts it first into a alcohol, then into an aldehyde, then into a carboxylic acid. Yep. Mixed function oxidase. So you get a formate out of that. Okay, so from this, you will get the molecules of water, and then the formate, and then also the product that you can see there. Yep. And notice you got yourself a double bond there now. So this product is called 2425 dihydrolinosterol. 2425 dihydrolinosterol. Dihydrolinosterol. Or excuse me, dehydro, I'm sorry. Dehydro, D-E-H-Y-D-R-O, linosterol. Now, in fungi, this enzyme is relatively different. if you compare the structure of CYP51s throughout, they're found in bacteria, they're found in plants, they're found in animals, they're found in fungi. The mode of action of these enzymes is very well conserved, but the proteins sequence very significantly across the kingdoms. This enzyme in fungi is inhibited by these groups of drugs we call the azoles. So ketoconazole, itraconazole, posaconazole, fluconazole, voriconazole, isuvaconazole, your mother-in-law azole. These drugs inhibit the fungal version of this enzyme, but don't inhibit the human one very well at all. So that's why these drugs kill fungi. They inhibit fungal ergosterol synthesis. Now, mutations that affect this first step can cause a genetic condition called Antley-Bixler syndrome. Antley-Bixler syndrome. Yeah, well, in humans. Antley-Bixler syndrome. And because cholesterol is an important signaling molecule, particularly in the development of the face, these individuals have facial abnormalities. They have skeletal defects. With this first one, the first reaction there. Yep. All right. You with me? Now, see that arrow on that double bond? And I've tried to let the, so you know the targets, I tried to put arrows where the targets are. So you don't look at the molecule and say, oh dear, what was changed? Okay, that double bond is going to go bye-bye. How do you reduce double bonds? You'd add hydrogens across them. So you use NADPH and you reduce it. And now, as you can see, you get the reduced molecule there. And that gives you the reduced version. And this is the C14 sterol reductase. C14 sterol reductase. But it turns out there's two different proteins that can catalyze this reaction. And at this point, things get really weird. Well, they're listed there. One of them is called the laminin B receptor. The laminin B receptor. What's the laminin B receptor? Do you know what laminins are? So laminins play a role in nuclear membrane structure. And this protein was first identified as a receptor for these nuclear lamins. So the laminin B receptor that is found just inside the nuclear membrane that binds it and provides structure to the nuclear membrane, that's where it was first identified. Then, when people put mutations in the gene, they discovered there were problems with cholesterol synthesis. Now, there's a second enzyme that can catalyze this reaction. It was thought to be the main player in catalyzing this reaction, this TM7SF2, which is found in the ER. But you need both proteins to be able to make enough cholesterol for the cell.
So they both can do it, but you need both.
Yeah. If you knock one of them out, you don't have enough cholesterol, even though the two proteins should be redundant. And mutations in that LBR cause Greenberg dysplasia. Greenberg dysplasia. Greenberg dysplasia. This condition is fatal before birth. And the bones of affected people look like they're moth-eaten. That's why this condition was first called calcification moth-eaten skeletal dysplasia. I like Greenberg dysplasia, it's much shorter. Yeah, and it's pretty serious. All right, steps 3 and steps 4. So steps 3, these two reactions are enzymatically coupled. And then they're repeated in reactions 5 and 6. So both sets of reactions require two distinct enzymes. There's a sterol C4 methyl oxidase, sterol C4 methyl oxidase. And that's why I have 2X underneath that, and then the other one 2X. So this reaction is going to occur twice. Yeah. So step 3 is step 3 and step 5. Step 4 is step 4, and it's also step 6. So step 3 and 5 are catalyzed by sterol C4 methyl oxidase. So as you can see, you convert that one methyl group there. And that methyl group, as you see, is going to be converted to CO2. You're going to lose it. And then next step, its brother, or sister, I guess, on the other side there is going to suffer the same fate. So you use molecular oxygen, NADPH or NADH, and you convert that to CO2. So you're going to oxidize that carbon. And in doing so, you convert that alcohol to a ketone. and then you reduce it back to an alcohol, and then you rinse, wash, repeat. And now the other methyl group will also be gone.
So the reason you do it twice is because you have two methyl groups.
Exactly right. Exactly right. And both of them are working on methyls attached to the C4 carbon. That's why they're called C4 methyl oxidase and C4 sterile C4 decarboxylase.
What are the other enzymes? Besides sterile C4 and methyl oxidase?
And C4 methyl, sorry, sterile C4 decarboxylase. Sterile C4 methyl oxidase, sterile C4 decarboxylase. And the sterile C4 methyl oxidase is encoded by the SC4MOL gene, and the sterile C4 decarboxylase is encoded by the NSDHL gene. That gene is X-linked.
Sorry, what was the other one that's not the methyl oxidase?
Decarboxylase. Decarboxylase. That's the reductase that's then used to convert the ketone back to the alcohol. That's a 3-beta ketosteroid reductase. 3-beta ketosteroid reductase. And it's encoded by this HSD17B7 gene.
3-beta ketosterol.
3-beta ketosteroid reductase. Because it's a carbon number 3, keto, and it's a steroid, you're reducing it.
Okay, I just so many words.
Now, this protein here, NSDHL. NSDHL, when you have mutations in that gene, you have a condition that is completely fatal in males. So you don't see males with it, but females will have one copy of the mutation. And they have what's called the child syndrome, CHILD syndrome, CHILD. Child syndrome. In males, it's fatal. You never see males with it. You only see females with it.
So it's with one of their exons.
Yep. Mm-hmm.
What happens?
They have limb defects. They have skin, little skin, so-called nevus, little black spots, freckles, but they're dark. They have limb rotation defects and skin lesions.
I don't know how more people are.
These conditions are not that common. They're pretty rare. But they're interesting because of the role that cholesterol plays in development even. They're interesting for that reason, I think. Okay. What are we doing? Yeah. Oh, time. Okay, next step. Now you are going to subject the molecule to, and this molecule is called, this molecule down here, this is called zymosterol. Yeah, zymosterol. Zymosterol. So you have an isomerase. And it is going to take that double bond, you can see with the arrow, and it's going to rearrange it. Right. That's step 7. Now that EBP protein is also X-linked, and mutations in it are only found in females. And they are called the Conradi. These people's names are long. Conradi. Hühnermann-Happel syndrome. That's mutations in EBP. Conradi, Hühnermann-Happel syndrome. This is also X-linked. So you only find this in females. Once again, lethal in males. Once again, we get skin and bone defects. And that's all we have time for today. Whoa. All right, we will, please bring this with you on Friday. We'll revisit this.
I want to rewrite it. Thank you. And did you bring our quizzes?
I forgot.
Okay, I'll follow you up.
I forgot.
Jenna, are you okay?
Oh, we so I showed you the picture. We did get some transfer. There was still a lot left in the gel. I think, here's what I think happened. I don't think ionic strength was high enough because the current was consistently low. And even though I let it transfer a good 2.5 hours, I think the transfer is just not, I don't think we got enough current because I think the buffer concentration was just too low. And the protocol that I used called for a lower concentration, but it just didn't work very well. I think concentration needs to be higher. Also, I got a call from the antibody people, and the antibody is delayed. So, well, we'll see. The storms apparently delayed shipping, so they're way backlogged. And we're in the hopper, but so I don't know what's going to happen with lab. We may just need to redo the gel and redo the blotting to make sure we get one, because I don't want to probe one and have it not work because we don't have enough protein on there. Sorry? Yeah, but that's easy enough. We've got columns, don't have to make columns. That we could do that, yeah. Not an issue. Not an issue. So, yeah, that's right. I hadn't thought of that. So, yeah, that we may need to do that. Okay. All right, Dave. Keep me posted and we will go from here. Okay?
Yeah, I will. Just so what slideshow did we go up to for this exam?
I think it was just the lipid synthesis. Cholesterol synthesis is not on this exam.
Okay, so we went up to lipids. Okay.
Wait, guys. No, lipids are not on this exam. No, it's just cell walls, Calvin cycle, gluconeogenesis, carbohydrate synthesis, glycogen, starch.
You had us include the first half of it, but you didn't put that on the plan.
Let me check really fast.
That's fine.
Okay, guys, there are no lipids on this test. No lipids, just gluconeogenesis, Calvin cycle, complex carbohydrate synthesis, cell wall synthesis, and that's it. No lipids.
All right, sounds good.
Okay?
All right, thank you.
You're very welcome. I will see you when I see you.
Oh, yeah, hopefully Wednesday, but
yeah.
All right, I'll keep you posted.
Okay.
All right, bye.
Bye.